Bismillahirrahmanirrahim. 970082 level, paper 4.2. This is the first video on this paper, which was examined in June 23. As you all know, this paper is for 2 hours, and you've got to finish this paper well in time so that you can revise it and see how you do not make any careless mistakes, because sometimes you just don't write the unit, sometimes you didn't read the word to the nearest whole number. All those mistakes you cannot make, because this is a very tough paper and the grade threshold is very high. It's a very difficult paper and you need to be very clear about how you're going to attempt it.
Now, please remember in paper 4 that you must read the whole question before you start attempting to do it, because you can easily make a mistake and write something which is also part of the next part of the question. So if you've written something in part A and you're writing it again in part C, well, then there's somewhere wrong where you're not writing it, because you can't be getting a mark for the same content. If you're writing the same thing, then you are not going to get marks in somewhere where you're getting it wrong. Glycolysis is a biochemical pathway that occurs in the cytoplasm of cells. In glycolysis, a molecule of glucose is metabolized to 2 molecules of pyruvate. The process is outlined. You can see all that as a very nice outline of it. Explain why glucose is phosphorylated at the beginning of glycolysis. Now, I'm not reading the whole question, but I expect you to read the whole question when you're attempting the paper. So why is glucose phosphorylated? The reason is glucose is activated or is made reactive, and then it becomes unstable and it cannot leave the cell, because please remember, glucose enters by facilitated diffusion through a channel protein. So when it becomes glucose 6-phosphate, that cannot move out of the cell, so it is sort of in a way trapped inside the cell, and energy level is increased and the concentration gradient is maintained. So if it doesn't leave the cell and it is going to be converted to pyruvate, so that means it will always be less inside the cell and more will diffuse in from the outside. So a concentration gradient is maintained. So it's only 1 mark, but you can even get this in another exam for more marks. So you must know this. So glucose is activated, becomes unstable, cannot leave the cell, the energy level is increased, because so much glucose in it, of course, will be converted into pyruvate, and of course, remember, 2 ATPs will also be made. Of course, we'll spend 2 ATP, but we will make 4 ATP, so a net gain of 2 ATP. So explain why glucose is phosphorylated at the beginning of glycolysis. Look at the mark and write at least 2 points so that you get your 1 mark. So just one use of the reduced NAD that is produced in glycolysis. What is the use of this NAD? So you could have written a lot of stuff. It's a carrier of hydrogen, or you can say provider of H ions and electrons. You cannot say provides H ions, because it provides hydrogen, so there is a proton and an electron. And used for the redox reactions and used to reduce ethanol, or you can say pyruvate. So it didn't say anything. It said one use of the reduced NAD that is produced in glycolysis. Now you remember, glycolysis occurs in aerobic as well as anaerobic respiration. So basically it is providing that hydrogen, which is used for then the redox reactions and used to reduce the pyruvate. So the pyruvate is going to be reduced. So everybody has to know this. What is the use of the reduced NAD? NADH. Reduced NAD means NADH. So it's a hydrogen carrier used for redox reactions and used to reduce the ethanol, or you could have even said reduce the pyruvate. Then name the type of phosphorylation reaction by which AT is made during glycolysis. Now that's actually a point of the syllabus. If you look at the syllabus, that's a very important point. This is called substrate linked or substrate level phosphorylation. Substrate link, or you can say substrate level phosphorylation. Then coming to the B part of the question, pyruvate can enter mitochondrion by active transport. So they're telling you this is a fact. Describe the main conditions that are required for pyruvate to enter the mitochondria. Now, what is needed for active transport is a very simple thing. It's 3 marks. You know that there's a carrier protein needed for that. In this case, of course, it's a SIM-port. It's a very special carrier protein called mitochondrial pyruvate carrier. But you might not know that. It doesn't matter if you don't know that. It's just a carrier protein got you a mark. The fact that ATP is needed got you another mark. The fact that oxygen is available. The oxygen is the pulling force which pulls the pyruvate into the mitochondrion. And the inner mitochondrial membrane is impermeable to pyruvate. So basically, you need a carrier protein, and there's a lower concentration of pyruvate outside the mitochondrion. So this is the mitochondrion, this is the outer membrane, this is the inner membrane. There is a channel protein here. So the carrier protein is there's going to be less pyruvate here. So there's a carrier protein, there's a lower concentration of pyruvate outside, the mitochondrion. So it has to be taken in by active transport. And active transport requires ATP, or you can say energy is needed. ATP or energy. Any of these two words would be correct. And oxygen has to be available. You see, oxygen is the pulling force. That's why it only happens, the pyruvate only enters the mitochondrion in aerobic respiration. It does not enter in anaerobic respiration. So carrier protein, lower concentration of pyruvate outside mitochondrion, oxygen should be available, ATP is needed, and the inner mitochondrial membrane is impermeable to pyruvate. Then coming to the last part of the question, there's a total of 9 marks, this chapter on respiration questions. Pyruvate is involved in the link reaction in the matrix of the mitochondrion. Describe the link reaction. Very direct question. Decarboxylation, dehydrogenation, reduced NAD produced, and formation of acetyl coenzyme. So very basic. You could have even done a diagram of it. I would say you just do a diagram. 3C pyruvate to 2C acetyl CoA. That's how I teach you. Acetyl coenzyme A. You could have even said acetyl CoA. That's acceptable. And then what is going to be happening? NAD is going to be reduced. So NAD, you put an arrow here. NADH. You got a mark for that. And then this is the dehydrogenation. And then of course also carbon dioxide is going to be exiting. So this is decarboxylation. Whether you wrote this or you do a diagram, you can even do a diagram. Some of you are very scared of doing a diagram. I don't know why. But it doesn't mean if the lines, you can do a diagram on the lines as well. You knew this. I mean, of course, I teach it like this, so maybe I know it and I'm very comfortable with doing this. So basically 3 carbon to 2 carbon. So the 3 carbon, 1 carbon is being lost as carbon dioxide, and that's called decarboxylation. So you could have done this like this and got your 3 marks for this. I will give you the points as well, written points as well. You could have said decarboxylation. You got a mark for that. But if you've seen the carbon dioxide exiting, you can even get a mark for that. And then dehydrogenation is the hydrogen being removed, NAD being reduced to NADH. So that is dehydrogenation. And then of course reduced NAD produced. You can even say NADH, that's fine. Reduced NAD produced. And the last point is formation of acetyl CoA. That's it, 4 marks, any 3 points, and you got your 3 out of 3. Question number 2a. In sexual reproduction, meiosis occurs to produce haploid gametes from a diploid cell. Explain the meaning of the terms haploid and diploid. Very basic. Haploid is 1 set of chromosomes and diploid is 2 or the complete set of chromosomes. Or haploid is 1 of each pair, 1 set of chromosomes. You could have said haploid is n chromosomes and diploid is 2n. Or you could have said haploid is half the number of chromosomes as diploid. Then it's a direct question again. B. Describe the behavior of chromosomes during the main phase of meiosis 1 in animal cells. So prophase 1, what is going to happen? Chromosomes condense, shorten, fatten. Chromosomes condense. Please do not say DNA. Chromosomes condense. Homologous chromosomes pair up, and crossing over may or may not occur. Then in metaphase 1, So this is here for prophase, and for metaphase 1, you would say homologous pairs of chromosome line up on the equator, and there's a random assortment of the homologous pairs. Then in anaphase 1, chromosomes, one of each pair of chromosome moves to the pole. Then telophase 1, chromosomes reach the opposite pole, and or the centrioles also reach to the end of the cell, and the chromosomes then decondense. So these are all the descriptions for this. Then the production of haploid gametes by meiosis also involves division of the cytoplasm. States the term used for this as cytokinesis. Cytokinesis. So cytoplasm divides into two, which is called, the word is cytokinesis. As you can see now, how do you write it? The wordings have to be very correct. Haploid is one set of chromosomes and diploid is two complete sets of chromosomes. Or you could have said haploid is n, diploid is 2n. But that'll only get you 1 mark because it's a very comparative statement. That's not going to get you 2 marks. So maximum of 1 if you do that. B part, prophase 1, chromosomes condense, homologous chromosomes pair up, and the crossing over may or may not occur. But of course, you could have given any 1 mark because there's 4 headings and 4 marks. Metaphase 1, divalent line up on the equator, independent assortment of homologous chromosomes takes place. Anaphase 1, one of each pair of chromosomes moves to the pole of the cell. Or you could have even said the end of the cell. That was allowed. Won't be allowed in AS. Telophase 1, chromosomes reach the opposite poles and chromosomes decondense. So these are alternative points which you could have given, and that would have got you your 7 out of 7. Then coming to question number 3, the sea brush, whatever, is a flowering plant that grows on the west coast of North America. Sometimes you see, you get very unnerved by reading these difficult names, so sometimes I just won't read them. And it's easier for you. I mean, I get a little unnerved, maybe you don't. The sea blush is a flowering plant that grows on the west coast of North America. Individual sea blush plants produce fruit that is either winged or wingless. Fruit. Investigations have shown that this characteristic is controlled by a single gene with 2 alleles. A dominant winged fruit allele and a recessive wingless fruit. So the dominant would be what? Can be AA or can be AA. This would be the dominant trait. And the recessive would be, of course, all small a's. Figure 3.1 shows a difference in structure between winged fruit and wingless fruit. Winged fruit and wingless fruit. So this is a winged fruit and that's a wingless fruit. And they've also given you a scale, and they both are nearly the same size. Wingless fruit 2 millimeter. A large sample of sea blush fruits was collected and their fruit wing characteristic was recorded. Name the type of variation. Well, they're only winged, or so it's discontinuous variation. Can't be continuous. Continuous, you have many intermediates. So name the type of variation. You just have to write discontinuous, and that would get you your 1 mark. Then it says different sea blush plants can have fruit with different colors. Suggest 2 ways in which a new fruit color could naturally occur in a sea blush population. Well, it could be a mutation, which will result in the formation of a new allele. And this would, of course, change the selection pressure. And maybe cross-pollination will start to take place. And this will result in new allele combinations. Because you see, a new fruit color has, maybe it's a codominant phenomenon. A codominant allele has suddenly occurred. So suggest 2 ways in which a new fruit color. So very easy. Mutation was a very obvious this thing. And how would it occur in a 2 ways in which a new fruit color could naturally occur in a sea blush? So mutation 1 mark, change in environmental or selection pressures, then cross pollination, or you could have said cross breeding or hybridization, and this would of course result in new allele combinations. Part 3 of the question, early taxonomists classified seed blush plants with winged fruits as a different species to the seed blush plants with wingless fruit. Early taxonomists. Since this time, evidence from observations and experiments has confirmed that the plants belong to the same species. Suggest 3 examples of the evidence obtained that help to confirm that these seed blush plants belong to the same species. Now, if you remember the syllabus, only then you can do these questions. The syllabus is very important because the syllabus said that you had to know that why would they be the same species. Basically, both types breed to produce fertile offspring. Both occupy the same niche. Both have same DNA sequences. These have all been done much later on now. DNA sequences were not there 50 years ago or 100 years ago. So have similar DNA sequences, or you can say same genes, or you can say the same genome. And then that results, because the DNA sequences are the same, so the same proteins or the same amino acid sequences are found in the proteins. So give 3 examples of the evidence obtained that help to confirm that these 3 blush plants belong to the same species. If they're the same species, only then they can interbreed to produce fertile offspring. So that's the definition of species. Then they occupy the same niche. Then they have the same morphology, physiology, and biochemistry. So basically, they're asking you to explain the definition of species. They have the same DNA sequences. They have the same protein or the amino acid sequences in the protein. Interbreed to produce fertile offspring, occupy the same niche, same morphology, physiology, biochemistry, any one of those words. I'm not saying you to write all 3 of them. Same DNA sequences, or you could have said same genomes, you could have said same amino acid sequences, or you could have said same protein. So when you read the question, be careful. Suggest 3 examples of the evidence that confirm that these belong to the same species. So basically, they're asking you the definition of species. Then coming on to the next part of the question, B, 5 marks, very direct question, very easy. Everybody should know that whenever there's such a question, natural selection occurs in populations such as populations of seed blush plants. Explain why natural selection occurs in populations. Now, this question has come in so many papers, and you can see the mark scheme is also very, very obvious. Populations can produce many offspring. Genetic variation occurs in populations. Mutations, or you can say sexual reproduction or random fertilization. So then competition for food, for mate, for resources. Then selection pressures, environment changes, so selection pressures change. Some individuals have advantageous adaptations, or they are advantageous phenotypes, or they have advantageous traits. So they survive. They have a selective advantage. And then pass on these advantageous alleles. I always say that you never write advantageous genes, because we all have the gene for blood group, but we have different alleles. So pass on their advantageous alleles, and the advantageous allele frequency increases. So whatever is an advantage to that organism, if that flourishes, then the others will die. Like I give you the example of giraffe. Once upon a time, there was short-necked and medium-necked and long-necked giraffes. Now, why have the short-necked and the medium-necked finished? Because now they don't have, their habitat is not suitable for them. So they have all become extinct, and only the long-necked giraffes are left. So the advantageous allele frequency increases. Pass on, the ones that survive pass on their advantageous alleles to their offspring. So all the offspring now will have long necks. So all the long necked giraffes still exist while the others have become extinct. Now there are some topics in which you should please memorize these mark schemes, because these come in every paper, and you can easily get your full marks from this. This was 5 marks. It's a very direct question. So natural selection occurs in populations. Why? So can produce, because all populations can produce many offspring. Then genetic variation occurs in populations. Then mutations can occur. Mutations can occur, or you can say sexual reproduction or random fertilization. All those points result in variation. Then competition for mates, for food, for resources. Then selection pressure changes, like the environment changes. There's drought, or there are certain insects which become extinct. So because then pollination will be affected. So selection pressures can change. Then some individuals have advantageous adaptations, or they have adaptations, or they have better phenotypes which are more conducive to this new environment. So those individuals will survive. Like I always say, in this generation, those children who are very computer literate and who can go on the YouTube and study will be success stories in life. They will not be dependent on any school, any teachers. Their parents will not have to fork out so much money. They need not spend so much money on petrol to go to school and come back, and they can just use the YouTube to study anything or everything. So selection pressure, some individuals have advantageous adaptations, they survive, and these will have a selective advantage. So they will pass on their advantageous alleles. Now that's why I've written it in red. Advantageous allele. The word is advantageous allele, not gene. And so the advantageous allele frequency will increase. So that which is an advantage, that allele will increase. Just like I gave you the example of the giraffe just a little while ago. Now coming to question number 4. Genetic technology uses many different enzymes and techniques. Restriction endonucleases are used in genetic modification. These enzymes occur naturally in prokaryotic cells. More than 3,500 different restriction endonucleases have been identified, and it is thought there are many more to discover. Name 2 domains that are a source of restriction endonucleases. Well, you know that there's in bacteria and archaea. So bacteria are where they are contained because they cut down alien DNA. So bacteria and archaea. That got you 1 mark. Now, of course, it's a very long question, a very direct question. Originally, the method used to obtain restriction endonucleases was to grow large numbers of specific prokaryotic cells that are the source of the enzyme, break open the cells and extract and purify the extraction endonuclease. These are also called restriction enzymes. The original method produced only a small quantity of restriction endonuclease and was not economical. The newer method for large-scale production is to obtain the gene coding for a specific restriction endonuclease, introduce the gene into E. coli with a promoter that allows the gene to be expressed continuously. The newer method increases the quantity of specific restriction endonucleases produced. Suggest and explain the steps needed to carry out the newer method for large-scale production of specific restriction endonuclease. And that's a 4-mark question, a very direct question. And that's very simple. You obtain the gene for that specific restriction endonuclease. How do you get it? You can get it from the mRNA. You can get it extracted from the genome of the E. coli bacteria, or you can even synthesize it from nucleotides, but that will only get you 1 mark. Then you PCR to make more copies of the gene. Then you cut a plasmid with restriction enzyme to give you sticky ends, and then you mix the gene with the plasmid using the enzyme ligase, and then you add this reformed or recombinant plasmid into the bacteria, and you add a marker gene to identify that bacteria, and then you culture these bacteria, and then you extract and purify the enzyme. So basically all the steps, and you have to give me any 4 of these steps to get the 4 out of 4 marks for this. Obtain the gene, or you can give me an explanation of how to obtain the gene extracted from, synthesized from the mRNA of the bacteria, or extracted from the DNA of the bacteria, or synthesized from nucleotides. So this will be an explanation of gene. Then PCR to make more copies of the gene. Then cut a plasmid with the restriction enzyme to give sticky ends and combine the gene with the plasmid using the enzyme ligase. then add plasmid to bacteria, then use marker genes to identify the bacteria with the recombinant plasmid. We don't want to culture all the bacteria. We only want to culture the one which have the desired gene that we have put into it. And they culture the bacteria using fermenter, then extract and purify the enzyme. And, part C, describe the advantages of databases for the study and use of restriction endonucleases. You see, data stores a large amount of amino acid or the nucleotide or the base or the DNA sequences, and you share this data amongst each other. Then you compare the amino acid or the nucleotide or the base or the DNA sequences, and then you predict a model, which could be the visual model of the structure of the enzyme, and then you identify restriction endonucleases to use, or to even discover new restriction endonucleases. So advantages of database for the study and use of restriction enzymes. So large amount of DNA sequences, then you can access or share the data quickly, then you can compare DNA sequences, then you can use to predict the structure of the enzyme according to the DNA sequence, then that is the amino acid sequence, and then how they're going to bond, where disulfide bonds will be formed, where hydrogen bonds will form, where ionic bonds will form, and then you identify the restriction enzyme that you would like to use. Then coming on to the B part of the question, electrophoresis is a technique used in genetic technology. Paper chromatography is a technique used to investigate the photosynthetic pigments found in chloroplast. Compare the similarities and difference between electrophoresis and chromatography. Well, that's a new question, and that hasn't come in any old papers, and that's for 4 marks. Now, as you can see, the mark scheme is any 4 from these. Now, the first one is a very important one. You can see it, separation role. Both are separating techniques and both involve movement of substances. Now, in the second feature, now I like this, when you do a comparison, you say feature and then you say electrophoresis and then you say chromatography. You can actually do this also on the paper. You could have easily done this on the paper and drawn 3 columns and done this. So molecule separated DNA, RNA, pigments. Then medium is a gel, is a paper or a glass or a gel. Both, and actually you can use a gel. Separated by electric current, solvent, separated due to charges or polarity or length or mass, and this is also by mass, so both separated by mass. Then visualization using dyes, ethidium bromide or UV light. Here different colors of pigments and dyes are not added, and they just move different distances. And in both you can also say dyes can be added if you're adding TLC. Then identification, use DNA ladder, known length of DNA, or use of DNA probe. Here you use the RF value, and in both use of references or known values, with which we have known values from previous experiments. So all these you could have given that in a line and writing it in a sentence, or you could have given it to me in the form of a table with feature, electrophoresis, and chromatography. So that completes this question number 4, and we will discuss the rest of the questions in the next video.